Now that you know everything you need to know about amino acids, we can move on and talk about the real thing, proteins. First of all, we need to clear something out. When you link together a bunch of amino acids, you don't find yourself in the presence of a protein, but only of what we call a polypeptide. Well, a protein is one or even more polypeptides linked together that has also reached a three-dimensional, biologically active, stable structure called native conformation. We can make the difference between monomeric or multimeric protein whether it's made of one or more polypeptide, but in either case it has to reach the native conformation to be properly called a protein. Now, since they are made of amino acids, we can say that every protein has an intrinsic directionality, with an aminic group to one end, the N-terminal end, and a carboxyl group to the other end, the C-terminal end. Even though only the 20 natural amino acids build the proteins, they can combine in pretty much infinite ways, and so the quantity of proteins is ginormous or something like that. The mere differences between amino acids aren't enough to explain all of this though, because, as I said in the last video, proteins are way more complex than that. We need to talk about the 4 different structures of a protein. What we call the primary structure is simply the sequence of the different amino acids that make the backbone of a polypeptide. This is only the beginning though. Things start to get interesting with the secondary structure, more or less long regions of the chain folded repetitively and regularly, forming the alpha helixes and beta sheets. These regions are linked together thanks to hydrogen bonds that are formed between the atoms of the peptide skeleton. Note that this does not involve the lateral chain R, and since there are a lot of this structure inside a protein, even though they are only made of hydrogen bonds, they end up making it much more stable. All the other parts of a protein that are not part of neither alpha helices nor beta sheets are called randomly wound regions. Moving on, we find the tertiary structure, which is formed through bending and folding of the whole molecule and is the consequence of interactions between the R groups of the different amino acids. 4 different kinds of bond can help to make this structure: the hydrophobic-hydrophilic interactions, the ionic bonds that form salt bridges, the hydrogen bonds, and disulfide bridges. Last but not least, we found the quaternary structure, which is composed of the mutual interactions, and by that I mean the same kind of bond that occurs in the tertiary structure, of the individual monomeric subunits that form a multimeric protein. This means that only multimeric proteins present a quaternary structure. The classic example of a multimeric protein is hemoglobin, a protein made of 4 different polypeptides that are linked together. This is the most abundant protein inside red blood cells, and is the one responsible for the transport of oxygen around the body, that forms a bond with particular region inside the hemoglobin called in group. With all of this in mind, you can start to understand why proteins are so complex. Aside from being really big molecules, they also form these 4 different structures and behave accordingly in completely different ways. The different interactions of the structures of a protein determine the native conformation, which allows us to categorize proteins in 2 big families: fibrous proteins, which are very long and with a big secondary structure, and the last sentence was better in my mind for some reasons. They usually play a structural role outside cells. Some examples are keratin, collagen or elastin, and globular proteins. They have a compact shape and represent the great majority of the proteins inside a cell. That said, I still covered only a fraction of what proteins actually are, but we'll understand all the missing parts in future videos, where we'll cover how amino acids know in which order they're linked together to form a protein, and the whole process of the maturation of a protein. As for the moment being, this is enough to move on with the next topic, which right now consists of the most famous molecules around, the nucleic acids, RNA and DNA.